Good evening, everybody, and welcome to the fourth in our series of get-togethers in celebration of the Department of Psychiatry's 75th anniversary. Welcome. A theme that binds our Diamond Jubilee celebrations is biopsychosocial innovations in caring, learning, and discovering. And it's that latter one, research, that we'll focus on tonight. My name's Yeates Conwell. I'm professor and vice chair of psychiatry and the moderator tonight of a discussion with 6 of our faculty members who are leaders nationally and internationally in their unique areas of research. You'll meet them in a minute when we play a 27-minute video in which each explains and reflects on what they do. Then we'll come together for a 40-minute or so panel discussion in which you all, the audience, have the opportunity to ask questions, which you can do at any time by writing those questions in the chat as they occur to you, the chat box below. And then when the time comes, I will field them for the panel. So please feel free to share your questions and thoughts. Those questions can be general, open to the panel as a whole, or you can address them to specific panelists with regard to the work they do in a specific kind of question. That's fine too. Before we start the video, let me offer a few brief reflections of my own. I've always been interested in aging and mental health, so I joined the faculty here in 1985, which was well before the subspecialty of geriatric psychiatry had been established here in the US, which was in 1991, actually. I came to Rochester, though, because the people here were well ahead of other institutions, even at that time, in the sophistication of thinking about care, teaching, and research into health and illness in later life. And I attribute that characteristic of the place in large measure to George Engel and the biopsychosocial model being deeply rooted in the culture here. So it helped those pioneers in geriatrics understand 3 fundamental principles of work with older people. First is that the strengths and vulnerabilities of that age group derive from biological, psychological, and socio-environmental factors. That may seem obvious to us now, but at the time, not so much. Second, that in each of these 3 domains, it has an independent influence on health, as well as through important interactions with each of the other of the 3 domains. So the impact, for example, of the environment on expression of biological systems, the psychological impacts of biological processes, normal and abnormal, and so on. So it's complex. And that third, only by including and attending to all of the 3 domains and their areas of overlap and interaction in our diagnosis and treatment approaches can we really deliver the care that our patients deserve. So research is fundamental to how we develop our understanding of this model and the populations we work with. Now, of course, these lessons are equally true for our work with children and their parents and their siblings, loners and those who are lonely in groups, people with serious mental illness, and those, including in later life, whose brain is failing them. It applies to everyone. So that's what makes our work both challenging, but also compelling and ultimately effective in healing. So with those thoughts, let's get to these exemplars of innovations in research and what they can tell us about the past and future of the biopsychosocial approach as applied in research. Thank you. Let's roll the video.
Hi, I'm Steve Silverstein, the Associate Chair for Research in the Department of Psychiatry, and I'll be talking to you tonight about biopsychosocial innovations in research on schizophrenia and related disorders. I'd like to start by mentioning Lyman Wynn, who had come here from NIMH and was the Chair of Psychiatry from 1971 to 1977, and continued to be part of the department for many years after that. Wyman worked on a number of groundbreaking studies, including the Rochester High-Risk Longitudinal Family Study, which led to important discoveries about the development of people who go on to develop schizophrenia. And he was also involved in the Finnish Adoptive Family Study, which was one of the major studies in the field, especially throughout the 80s and 90s, revealing information about gene-environment interactions in schizophrenia. And in addition to that, he did a lot of work and wrote a bit on psychological approaches to treatment of schizophrenia. His work really exemplified the biopsychosocial model. Another major figure who was here at that time was John Strauss. He was here as the Director of Clinical Psychiatry Research from 1972 to 1977. And he did a number of very important studies that helped to define the modern diagnostic criteria for schizophrenia and to look at differences in outcome in schizophrenia throughout the world as part of studies with the World Health Organization. One of his other innovations is he was really interested in the narratives that people with schizophrenia have about themselves and in the subjective experience that patients have. So he was really exemplifying the biopsychosocial model and using it in his work long before it really became a popular concept in schizophrenia treatment. Another leader who was here at around that same time, going into the 1980s, was Rue Cromwell. Rue is a pioneer in experimental psychopathology, or using experimental methods from psychology to understand the mechanisms of schizophrenia. And he did many studies in patients, but also unaffected first-degree relatives, to try to understand what is the vulnerability to schizophrenia, what does that consist of? And he wrote many books and published many papers and mentored many people who went on to distinguished careers in research, including Will Spalding, who was here as a postdoc from 76 to 79. In 1979, Harut Babigian became the chair of psychiatry, and one of his major projects was the Monroe-Livingston Demonstration Project, which showed that a capitated payment system could lead to significant increases in the quality of care for people with schizophrenia and to reduce hospitalizations. And it was during his time as chair that Strong Ties opened, which later became a major site for research. In 1993, Eric Kane became chair of psychiatry, and at around that same time, Marvin Hers came from the University at Buffalo to introduce a schizophrenia research focus at Strong Ties and at the Rochester Psychiatric Center, where also 2 units were set up, including one that I helped to run and study the outcomes of. Starting around 1994, a research group was formed here in addition to what Marvin Hers was doing, which is studies on service delivery and relapse prevention. This group consisted of all young faculty. We were doing studies at Strong Ties and at RPC, focusing on many issues, including brain function and to treatment of schizophrenia. In 1995, Steve Lamberti initiated Project Link, which was the first forensic assertive community treatment program in the country. And this, he later on went on to receive an NIMH grant, the first one ever, to study that model, which is now being used in multiple states. And Steve and Rob Weissman are currently studying the effectiveness of that model in Minnesota. In 1997, we organized here, a group of us junior faculty, the first National Conference on Psychiatric Rehabilitation of Schizophrenia. And that was something that was happening as that treatment model was taking hold at Strong Ties and RPC. From 2005 to 2016, under direction of Steve Lamberti, our schizophrenia research here joined the NIMH Schizophrenia Trials Network, and we were involved in a number of studies that were the largest at the time of these interventions, such as newer antipsychotic medications and cognitive remediation. In 2017, Ben Lee became chair of psychiatry, and we opened the Young Adults Clinic, focusing on people recovering from a first episode of psychosis in 2019. A year later, that site became part of the NIMH Early Psychosis Intervention Network, or EpiNet. At around that same time, I came back to the university, brought some people with me, such as Brian Kane and Judy Thompson, and soon after that, Joy Choi joined the department. And we are now underway with a number of studies of brain function and structure, retinal imaging, cardiovascular issues, and other cognitive issues, including treatment of some of the cognitive impairments in patients. Just recently, in fact, this spring, we're opening a clinic called Strong Foundations to treat people at high risk for a psychotic disorder. And I'd like to close by saying that as it becomes clear that schizophrenia is not simply a brain disorder, but it's a multi-system condition that involves the endocrine system, involves neuroinflammation, involves altered metabolism. It's been very important to collaborate with other departments at the university with experts in those areas. And we've been really increasing those efforts in the last couple of years, and we'll be continuing to do so in the future.
Hi, everyone. Today I'm going to share some historical perspective on past research in our department, focusing on one of our most notable faculty members, experimental psychologist Robert Ader, PhD. By way of background, early in the 1900s, physiologist Walter Cannon became arguably the first researcher to apply rigorous scientific methods to demonstrate that when an animal is confronted by a threat, the sympathetic nervous system will become activated. This results in an acute stress response and numerous changes in the body, including accelerated heart rate, increased blood flow to muscles, and decreased appetite, what we know as the fight-flight response. For decades following Cannon's findings, however, it was believed by immunologists that the immune response, unlike any other systemic response, somehow escaped this regulation by the brain. And this may have been in part because much of the science of immunology was discovered in vitro, in the test tube. However, there began a groundswell of experiments round about the 1960s, continuing to today, from groups of scientists all over the world, that irrefutably shows that the brain and the immune system are intimately connected. These include the seminal finding that there is sympathetic innervation of lymphoid tissues. This was the groundbreaking work of David and Suzanne Felton in the 1980s from our own Department of Neurobiology and Anatomy. Psychosocial factors and stressors influence immune responses. Lymphocytes express receptors for numerous hormones and neurotransmitters. And finally, immune responses can be behaviorally conditioned. This last important point began with the work of Bob Ader, who was a faculty member in our department for 5 decades. In the 1960s and early 1970s, Bob was doing some conditioned taste aversion studies, pairing a novel taste, saccharin solution, with the noxious and immunosuppressive drug cyclophosphamide. He observed that when he stopped giving the rats cyclophosphamide but continued to give them the saccharin solution, not only did the rats avoid drinking the saccharin, which was evidence of clear taste aversion, but some of the animals died. The magnitude of the avoidance response of the rats and mortality were correlated with the amount of solution consumed. Bob quickly realized that he needed the help of a seasoned immunologist on further experiments. He found the perfect collaborator in immunologist Nicholas Cohen, PhD, now Professor Emeritus. They paired a solution of saccharin in water, the conditioned stimulus or CS, with injection of cyclophosphamide, a noxious drug that's highly immunosuppressive, to induce illness in rats. Saccharin plus cyclophosphamide also induced immunosuppression as expected, as evidenced by reduced antibody titers to antigen injection. When they continued to expose rats to saccharin in the absence of drug, they observed significantly diminished antibody responses compared to animals who had been previously conditioned but did not receive further exposure to saccharin. This experiment became clear evidence that one could behaviorally condition changes in immune response. In other words, the brain was directly regulating immunity and possibly mortality in a situation where no toxic drug was around. The coalescence of Bob and Nick's elegant experiment, along with the work of others, led to a rather interesting period of time with back and forth volleys between hardcore immunologists who raged against the growing body of evidence and those who were carefully exploring the science of mind-body medicine. Sometimes it got downright nasty. Bob kept the science moving forward with the establishment of a new journal called Brain Behavior and Immunity in 1987, and he was the first editor-in-chief. He formed the Psychoneuroimmunology Research Society in 1993 and was its first president. He had a long and well-honored career at the University of Rochester. Bob retired in July of 2011, and he died in December of 2011. He was my very dear mentor and very good friend. Thank you.
I'm Tom O'Connor, and I'll be talking about the biopsychosocial model as we apply it in our research in the Wynn Center for Family Research. A central idea and core hypothesis in our research is that early exposures and experiences may have clinically significant, persisting, and/or privileged impact on health and development. This is an idea that's been rattling around for some time. What's innovative now is our increased ability to formalize and qualify these hypotheses and dig in deeply to understand the causes and effects with techniques from behavioral science, genomics, immunology, and other advancing fields. Development does not begin at birth, and so in our program of study, we start in the prenatal period, measuring exposures and effects directly and indirectly. Over 20 years ago, working with colleagues in the UK, we provided some of the earliest evidence that anxiety and stress in the prenatal period can have lasting impact on neurodevelopment and somatic health outcomes in children. That finding has since been replicated many times in many countries using a diversity of methods. That work said something important about the role of early exposures and experiences and was equally notable for what it implied about the timing of preventive interventions. We've since expanded that work to the placenta and have made the argument that not only pathologists, but also psychologists and other practitioners of health research have a reason to consider the placenta in our studies of health and development. Our studies of human development from cradle to grave do need to start well before the cradle starts rocking. In our research, we study risk mechanisms because they provide the strongest clues about causal effects and can offer clear directions for targeted treatments. Stress exposures deriving from family and parenting conflict, trauma, and even environmental chemicals are of concern for mental health and for the associated biology and stress response. That's because stress physiology is related to mental health and illness as well as somatic health, that is immunology, endocrinology, and metabolism. Indeed, one of the most studied stress systems in the human body is innately mind, brain, and body, and chemical messengers created by that system can affect cell types throughout the body. In this area, our work with colleagues at the Eastman Institute for Oral Health has helping to demonstrate how biological, psychological, and social stress is important even for oral health and early onset caries, one of the major public health concerns in children. Also, our work with colleagues in pediatrics demonstrated for the first time that quality of parent-child relationships, as rated by independent observers, predicted antibody response to vaccination. We need to be continually sharpening our skills for advancing the construction and definition of what bio and psycho and social mean, and of course their integration. For example, our examination of bio now includes neuroimaging, the gut microbiome, immune mechanisms, genomics, using techniques and levels of analysis not anticipated in Engel's seminal paper published decades ago. Equally, the social determinants of health and matters of social and economic justice are different now and continue to change and require scrutiny in our clinical research. Clinical research supported by the Wynn Center for Family Research supports this ongoing mission. The work that we do isn't restricted by departmental or disciplinary boundaries. Our program of clinical research at the Wynn Center, which fully embraces the biopsychosocial model, involves collaboration with clinicians and scientists in pediatrics and neuroscience, obstetrics and gynecology, microbiology and immunology, the Eastman Institute for Oral Health, Public Health Sciences, Nursing, Environmental Sciences, and Biostatistics. The significance of family research has not changed, but certainly many of the questions and techniques that we use have changed. I mentioned in the previous slide our work on family research in children's immune health. To that, we have a focus on how caregiving may modulate early stress physiology that may compromise later health and development. More recently, we've added health economics to demonstrate that sensitive early parenting predicts not only behavior and mental health, but also cost.
I've actually been here since 1989. I did my training here and my fellowship. And I started focusing on Alzheimer's disease in the early 1990s. Back then, Alzheimer's disease was really just the clinical part of Alzheimer's disease. That is, that you had significant symptoms. But we understood quickly that you don't wake up one day having Alzheimer's disease, that actually it's a long process. The plaques and tangles are building up in your brain, and that happens maybe over 20, 25 years before you have any clinical symptoms. The disease starts with pretty mild symptoms, usually in short-term memory, that we have identified as a stage that we call mild cognitive impairment. Impairment in short-term memory. There may be slight impairment in other areas of cognitive function, but no real functional impairment. You really are able to manage your life, albeit with more effort. That is then followed over 3 to 5 years with a transition to the more classical clinical form of Alzheimer's disease, typified by the mild stage, the moderate stage, and the severe stage. When I think of Alzheimer's disease and I think of the biopsychosocial model, I feel that we have the biology of Alzheimer's disease fairly well understood and fairly well typified. What we often overlook is that there is also a psychological or emotional component to Alzheimer's disease, and that is that just about everyone that develops Alzheimer's disease, and even in the earlier stages, the preclinical stage and the prodromal stage, or the mild cognitive impairment stage, we see a significant amount of emotional burden. That is anxiety, irritability, depression, apathy, psychosis, agitation, and aggression. And those behavioral symptoms vary depending on which stage of the disease you're at. In addition to the behavioral symptoms that often are overlooked, we have the social component, and that is you lose your functional independence and somebody needs to step in. So every patient that develops Alzheimer's disease has multiple caregivers. Family caregivers most often, the elderly spouse, the adult daughter-in-law, the paid caregiver, the adult son. So for each and every patient that has Alzheimer's disease, the trickle-down effect through the family is tremendously substantial. At the Alzheimer's Disease Care Research and Education Program over the past 30 years, we've focused on intervention studies, many of them now targeting the underpinning of the disease, the amyloid dysregulation, the tau dysregulation, the neuroinflammation, and the impact from damaged neurotransmitter systems. But in addition to that, we focused on the behavioral aspects of the disease, the neuropsychiatric symptoms, and on the social aspects of the disease. And this is what I look forward to speaking to you more about tonight.
Recognition that individuals are embedded in thick social networks, and those networks influence health, is a key insight from social science research. Social networks are the natural relationship systems in our communities and workplaces. They impact health through the dissemination of norms, such as likelihood of using or abusing substances, support giving and receiving, and access to resources, including health services. In a study of the peer friendship networks across 40 high schools, our team found that students at high risk for suicide cluster together and are less connected to other students and adults. My colleagues and I are developing 2 intervention strategies to build protective factors into the fabric of social networks. Our Above the Influence of Vaping program illustrates the first strategy. 8th grade students identify influential members of their friendship groups, and those diverse key opinion leaders are taught resistance skills, and they disseminate those skills to peers using science-based prevention campaigns. Our work shows that students closest to trained peer leaders are more likely to resist or stop vaping by the end of 8th grade. With R01 funding from the National Institute of Drug Abuse, we're testing the program's impact into high school in 20 secondary schools. The second strategy is illustrated by our partnership with the U.S. Air Force to create the Wyman Connect program aimed at reducing suicides, currently the leading manner of death in the Air Force. Women Connect is the second type of network intervention in which all members of natural organizational units participate in group intervention. Women Connect strengthens two suicide protective functions of networks: stronger connections for support and belonging, and a shared culture that unifies members around healthy practices, including those that give meaning and value in life and work, and attaining emotional balance. In a randomized trial funded by the Department of Defense with 1,500 U.S. Air Force personnel in training, Women Connect reduced suicide risk and depression and cut by 50% the likelihood of corrective job training. Women Connect strengthened group bonds with the greatest impact for personnel who began more isolated and at elevated suicide risk. Additionally, the findings validated the underlying network health model by showing that the impact of Women Connect on increasing cohesive healthy units was an active mechanism reducing suicide risk and depression. We're now working with the U.S. Air Force to develop their personnel as trainers and expand the program force-wide. Taken together, our work suggests that network interventions may be critical to making prevention programs ecologically valid and have more sustained impact by becoming part of the relationship systems in which groups are embedded. Thank you.
My lab is called the HOPE Lab, which stands for Helping Older People Engage. And we study and develop innovative programs to help people stay connected and feel less lonely as we age. Well, we know that social connections are essential for health and well-being at all ages, and that social connections are something that are malleable throughout life. So even though loneliness doesn't increase with age, things like functional impairment and pain do increase with age. And we may not be able to always change those things, but what we can always change are our social relationships. It's something we can always improve. And so being able to intervene on social connectedness gives us a range of ways to promote health and well-being at all ages. We know that feeling isolated and feeling lonely leads to poor mental health, poor physical health, declines in physical functioning, declines in cognitive functioning, as well as suicide ideation, attempts, and mortality from all causes, as well as suicide deaths. The research has shown us that social connections that create a sense of caring, of contributing, and of belonging to a community have those health effects that promote health and well-being. And so now a project we're doing is looking at just that. We're looking at the benefits of being a volunteer through the AmeriCorps Seniors Program. And so we are taking folks who are willing to be in this project and randomly assigning them to either be an AmeriCorps Seniors volunteer or to do a project called Life Review, which is a evidence-based program for increasing well-being and reducing depression. And we want to compare these two and see what's the most effective strategy for reducing loneliness. And as well, I want to tell you about the Rochester Roybal Center for Social Ties and Aging Research. And we are able to develop and test a portfolio of programs for loneliness that are tailored to the unique needs of people who are caring for a loved one with Alzheimer's disease and related dementias. Individuals caring for a loved one, by definition, have a lot on their plate. And so the programs we may need to develop and deliver should fit the demands of someone managing caregiving and also fit the unique barriers and priorities that those individuals face. I want to tell you as well about a neat collaborative project my lab was able to do with individuals in the computer science department here at the U of R. We were able to adapt Dr. Ehsan Hoke's intervention that uses affective computing to create an online social communication coaching program for older adults. So we know that one contributor to feeling lonely or having unsatisfying relationships can be challenges with communication. So for example, being able to communicate with a doctor or an oncologist is a very different type of situation than many of us have encountered before. You might think as well about the context of moving into a senior living community where you might need to make new friends, and that's the first time you've done that in a while. Coaching can be a useful intervention for some people to either brush up on those skills or gain new skills. And what you see on the screen is the avatar that individuals are able to have a conversation with. And it's a back and forth. It's fueled by natural language processing and affective computing. And it's automated, and an older person can have a conversation with this avatar and then get feedback on how they do with things like eye contact or speaking volume. I'll tell you about one more really neat project that we did during the pandemic. And this was a pilot project looking at potential benefits of robotic pets. And the person who adopted this robo puppy also really liked magic and actually created a magic show for other residents that use the puppy. And so these robotic pets, they do sense light and movement, and they will bark or purr, things like that. And in the CLC, they were able to foster connections between people using those robo-pets. And finally, I want to just say that I study connectedness, and I want to be connected with anyone who's interested in talking more about this. So please scan the QR code to learn more about the Health Lab, and please stay connected.
Excellent. Thank you, everybody. Let's gather around the table here. And again, I'd remind all the folks in the audience to please put your questions and comments into the chat box, and we'll try to address them all in the 40 minutes or so that we have remaining. And again, they can be very general, or they can be very specific to a particular panelist, to try to understand more and in greater detail about the nature of the programs of research represented here today. It's really fun to hear both the history, the context of the work that you all do, and as well as the details of the research. Let me start while people are populating the question box there with just some ask your general reflections on it's kind of a controversial topic in a way. It's the question of whether the biopsychosocial model is a theory, how it actually applies to research. It's been criticized in some quarters for not being a theory, given that theories are so important to guide our research questions, our measurement, our methodology, our interpretation, and move the field forward. And if the biopsychosocial model is not a theory, I guess that's for discussion, is it still useful somehow for generating hypotheses and rigorously testing them? Can it effectively move the field forward? What kind of thoughts do you all have about that?
Well, I think that the model produced a tremendous amount of research, back in the day, but maybe it's a little old-fashioned or antiquated at this point in time. But Hippocrates back in 400 BC was talking about basically mind-body medicine. And I think the biopsychosocial model just sort of gives you a better umbrella for fitting in all of those studies that we've all been doing. But, I think it's just guidance. It is not the theory itself. That's okay. They're good words.
One thing we should probably give it credit for is really helping develop a common language certainly across our medical center for the integration of these different systems. And I think it has definitely contributed to the really collaborative nature of our medical center, and again, a sort of entering research together with a common language. So I think in that sense, a single theory or a more narrow, specific testable theory couldn't do that, I think, in the way that this kind of framework and orientation can do.
Yeah. Reminds me of a notion that, what was it? Somebody said everybody has it was the theories are like toothbrushes. Everybody has one, but nobody wants to share it with others. They want to just use their own.
Well, I don't really think that's true of scientists, do you?
Tom, you were going to say something?
Yeah, I don't think it's a theory, but it's a very useful framework. As Jan and Peter said, we've got lots of useful other frameworks that are in that sort of domain: social determinants of health and social neuroscience and psychoneuroimmunology. It's not like we try to think about studies to falsify the biopsychosocial model, right? So falsifiability is one of our key sort of strategic approaches. It wouldn't fit that, but as I said, it's a terribly useful framework for developing theories.
So, Yeates, let me give you an example, though, in my field, and that is that I think that reductionism still, reigns. I don't know that it rules, but reductionism reigns. And, I do a lot of intervention research with pharmaceuticals, and we see a strong movement there for all kinds of reasons to move to more narrow outcomes, biomarkers. So let's talk about Alzheimer's disease and all the controversy that has happened with a humanized monoclonal antibody that was approved by the FDA based on its ability to reduce beta amyloid, the protein, toxic protein that builds up in the brain. But there is no kind of agreement that that is closely connected to clinical outcomes. So we see a lot of that, and then the expansion to see that actually translate into a meaningful clinical difference, which I think then bridges, not only the changes in your brain, but your ability to think, your ability to function, your ability to relate to others, the burden that is on your caregivers, and your own, independence, your own autonomy. So I think that if we don't have a reminder that we need to look broadly, then we run the risk of kind of heading into kind of more narrow camps, and that's very much the impact that I think on my thinking from kind of growing up, medically speaking, in the department.
Mm-hmm. Okay. Many of you deal with very specific research questions, and yet you also deal with broader areas of inquiry and seek to put those specific questions into practice to make them meaningful. I mean, a study of a particular neurochemical, and it's or back to you, Tom, the notion of epigenetics and the impact of the environment that has such long effects. How do you balance those things? As a scientist, how do you decide what's the narrow question and then to make sure that it's applicable in a broader context?
Well, tragically, Yeates, one study can't answer all the questions we have. So we have to pick and choose what is at the fringes, what's a bit exploratory, what's a bit foundational. And have colleagues who know enough bits on the fringes of our own knowledge that we can do useful transdisciplinary research.
Mm-hmm. Mm-hmm.
In the vague answer.
We come from a kind of a unique environment here with the biopsychosocial model having influenced so many of the people that influence us. Do you find when you venture in your collaborations outside of the University of Rochester that there is a similar nuanced appreciation of the way that the biopsychosocial model can inform research? Are there any examples of that anybody can give? Or do you feel like a duck out of water when you go to collaborate with others?
Well, over the last many years, we've had the decade of the brain. We've had NIMH investing most of its money in genetics and brain imaging research. We have lots of pharmaceutical ads on TV. You never see a commercial on TV for how it's important to study the social determinants of health. And so, certainly at most conferences, and you can read journals, and, you see that there are certainly other people outside of Rochester doing work at the intersection of biological, social, and psychological factors. But I have to say, it seems to me that the field of psychiatry, as it's been for many years, at least since I got into it in the 80s, has been primarily focused on the biological. And that's really why I think the biopsychosocial model is still important, even if it's not a formal theory. You're not going to make it into a computational theory with equations and all that, but it suggests that you should be looking at a number of different influences on a person, and that those influences are important. And really, when you study those things, you usually find some significant effects, as has happened here many times. When Lyman Wynn was here, he was looking at children born to mothers of schizophrenia who were adopted away. And some of them went to very healthy families. Some of them went to very psychologically unhealthy families. And, the rate of schizophrenia in those two different groups of kids was very different. So the genes and even the brain you're born with doesn't determine everything. In fact, other things determine quite a bit. So yes, I sometimes feel like a fish out of water, but there's enough of us who care about this that makes it bearable.
Mm-hmm. Mm-hmm. Okay. The dominance of the biological in psychiatric practice and training and research that you're referring to underscores the importance of the biopsychosocial model as kind of a counterpoint to reductionism in medicine and research, as I think you're referring to. But in a similar way, the three components, biological, psychological, and social, that compose the model are a bit, that suggests a certain kind of reductionism itself until you look at the interactions between them and recognize that all need somehow to be incorporated into your models and subjecting that study. So the biopsychosocial model is more than those three things, right? And I think you've all given some examples of that. But it poses real challenges, doesn't it, in terms, what are some of the methodological issues that challenge you all in being biopsychosocial in the work that you do?
I think one of the things is that it's very important to have the collaborations that you need to do that kind of research, right? So I'm an immunologist by training, but I spent most of my life working with Bob Ader, who's an experimental psychologist. And I've had collaborations with Tom O'Connor since I think he walked in the door. So you just have to find the right team of people. You can't go it by yourself. It can't be done.
It's team science.
It's team science.
Yeah. Maybe Kim, if I could draw you into this, your partner. Who are your partners in research? You think that help you cross that from one to more?
Oh, well, definitely Dr. Kathy Hefner, who I know is listening, would definitely be a close colleague. We're able to do the work on caregiving together. And I guess that makes me think about another piece of the biopsychosocial model that isn't as explicit, but coming to your question, Yeates, about what's challenging, I think it's the idea of development and trajectories. And when we really ground ourselves in the biopsychosocial model, we have to acknowledge that there's a time course. And when we think about Ben Chapman's work, looking at the early influences of things like poverty on our developmental trajectories, those social influences are so important.
yet so challenging to measure. And then when we're trying to intervene and help people, they've had a lifetime of some of these exposures that change their health. And so I think that's a big challenge when you're trying to, say, do an intervention study when you can only change so much.
Mm-hmm.
If I could just, jump off of what Kim was saying, I think one of the challenges, the really social part of this model, and I think the current era we're in, revealing and recognizing the way that adverse histories become internalized, become part of biology, become part of psychology, affect health. Unfortunately, what we have really often to offer in terms of interventions is driven by a patient care system that really pays for encounters after people are ill. And I think one of the greatest challenges is how do we really focus more of this, attention, resources onto these powerful social factors that have dramatic effects on multiple dimensions of health and well-being. And I think, being in a medical center, I think there's an excitement of being in the applied arena, but also I think we're aware of how much the medical business system sort of drives the paradigm for what we have to offer really our communities. And there are some exceptions, or New York State, other systems are investing in prevention, but the overall the balance is quite skewed towards really post illness intervention.
Mm-hmm. Great. There are a number of questions that are coming in here, and let me just be sure to try to get to those. Here's one I think maybe to you, Jan. This is from a dear colleague of ours in Atlanta, Walid Nassif, who says psychoneuroimmunology is quite developed here in Atlanta under the leadership of Andy Miller. But he rarely mentions Bob Ader. Is there cross-center collaboration for this fundamental topic?
Can you spell ego?
Oh, back to the quote about theories being a toothbrush, right? I mean,
yeah, that is surprising. That is surprising. Yeah. Andy Miller is certainly a force in the field and has been for many, many, many years. I think in every kind of little fledgling field, there is though sort of a battle of, a battle of egos. So there you have it.
Yeah. Well, I know many of you do, as I referenced before, have collaborations across the world, and they are very, very productive and important ones.
Yeah.
There's a question here. Is there work being done around critical times when psychosocial factors have an impact on biology and when it does not? So when social networks are more protective and when they are not? And some version of that question could be asked for each of you.
Well, that sounds like something Tom should have.
I was thinking, Tom, does your work get to sort of the notion of sensitive times in development when different of the factors in different domains have an impact?
Yeah, we do. Gosh, that's a long story. I think that the general take-home message is to if we could add another letter, I think 3 seems to be the max, right? Biopsychosocial, brain and behavior immunity, psychoneuroimmunology. No one's gone to 4, but if we could go to 4, we would say a developmental model of biopsychosocial, so that we could incorporate developmental timing, which of course has an influence on all of the things of interest to the B and the P and the S, whether it's gene expression or in utero exposures or others. So that's a general principle. Whether there are particular periods of sensitivity, well, that is a very long story. I think there are some who've, I think, over-egg the batter a bit, maybe, in sensitive periods, although clearly there is some evidence for it. But I think that the general notion is if we're interested in development of the brain, then, for example, one very easy implication is that we shouldn't start at birth because the brain starts developing prior to birth, which is why we start prenatally. So as one example of how we would take development seriously in the biopsychosocial approach.
Mm-hmm. I wonder, Anton, if you could speak to that. I know that you referenced the development of symptomatic Alzheimer's disease as a biological phenomenon begins well before manifestation of clinical symptoms, right? Is there what do we know about that developmental process and vulnerabilities that may predispose to adverse course and outcomes?
What we know is that it's complex. I mean, we know, for example, from the study of nuns here in the U.S. that certain members of the order that had greater language capacity, language skills at entry into the order, which usually was late teens, early 20s, that those individuals appeared to have lower risk of developing Alzheimer's disease, maybe 50 years later. We know that lower educational attainment increases your risk. We know that sedentary lifestyle increases your risk. We know that limited social circles increase your risk. We know that untreated depression increases your risk. So there are things that we are aware of that you can do through your lifetime, like lifelong learning, regular exercise, thoughtful diet, social engagement, and basically, even your value system may have an impact on bending the curve. Nothing seems to be absolutely protective. There's nothing, I've seen people that, have exemplary lifestyles and yet get Alzheimer's disease in their young 60s. And you hate to think about what it would have been if they didn't live such an exemplary, quote, brain healthy lifestyle. But we know that these things matter, and that it's a very broad, it's not just one thing, and you can't reduce it. For example, if you look at dietary issues, that consuming seafood seems to be modestly protective, but if you just take fish oil, that doesn't seem to do it. So there's more. You can't kind of be reductionist about it.
There is a specific question here to follow up for you, Anton, from the audience. How early can CSF analysis detect the onset of Alzheimer's disease? You spoke of 25 years. Is that CSF or biopsy data?
So no, it's biomarkers. Basically, biopsies are so yesterday in a way. Those are receding, thankfully. Whenever I took out the Black & Decker and offered to do a core sample, that was always the last time I saw that patient. So now we can actually look at things like you can do CSF analysis. You can look at A-beta, and we're continuously learning more and more. I mean, just this week, it was pointed out that basically not just looking at A-beta 40 and 42, which are the most often discussed subspecies of beta amyloid, but looking at even shorter chains, so A-beta 37, 38, increases your power and your sensitivity to find people early that are at risk of developing Alzheimer's disease. So you can look at CSF, you can look at amyloid PET scans, tau PET scans, you can look at sensitive MRIs. That comes later, much closer to onset of symptoms. But intriguingly, blood and plasma biomarkers are becoming quite sophisticated, and they can pick up subtle changes long before you have any clinical symptoms. So if you're going to try to pick up the first changes that we see 20, 25 years earlier, and how do we get at this? Basically, much of this comes from our studies of dominantly inherited Alzheimer's disease, where families have a very similar age of onset. So members of a family have a very similar age of onset. So you can kind of predict that a child of someone who has an onset of Alzheimer's disease at age 55 is likely to be hit around that time. And as early as age 30, we can pick up the first changes in PET scans and CSF. We're not quite there yet with blood samples, but we're getting there. And then, of course, Yeates, what do we do with that once we have that sophistication and ability? Do we go out and find all these people and tell them long before that they have this risk? And what's going to be the impact of that if we don't have well-defined interventions to offer at that point?
So there are important ethical dimensions to the biopsychosocial implications of illness. Speaking of biomarkers, Steve, there's a question about your Center for Retina and Brain. Can you say a little bit more about that? And how does studying perception, such as visual and auditory processing, help us better understand and treat schizophrenia?
Yeah, well, that's a good question. For the center, the idea, of course, is to not only study schizophrenia, but to look at retinal and other eye markers of systemic disease outside of ophthalmology, because it's becoming increasingly clear that, especially the retina, where there's neurons, very often the changes you see there are similar to the changes you're seeing in the brain, such as loss of neural tissue, loss of vasculature, inflammation, and things like that. So it's in neurology and cardiology and several other areas of medicine, people are studying the eye more because it can be non-invasive, very fast, inexpensive, and you can learn a lot about what's happening in the brain or other organs. So at our center, which we only opened about a year ago, we are looking at retinal biomarkers of brain and cognitive function in schizophrenia, as well as in people with heart failure who are undergoing LVAD treatment, which should improve blood flow to the brain and should improve cognition. We want to see if you can look in the eye to see if who's responding to that. We're also looking at college football players before and after a season and looking at the changes in brain function and brain structure related to concussions and other head impacts. And again, what you can see in the eye from that. And generally, though, with schizophrenia, I should say that one of the strongest predictors of who's going to develop schizophrenia is poor vision in childhood, and also the onset of perceptual visual distortions in adolescence and early adulthood. These are very understudied. And so essentially, these are the kind of things that we're studying at the center now, how to understand how the eye and the visual system may tell us about the mechanisms involved in schizophrenia and some other brain disorders.
Thank you. One of the issues that's come up here is around the COVID pandemic, which forces so many questions and concerns around biological, psychological, and social influences on health. And it also exposes disparities in health access and outcomes. So the question that came up here is, can the biopsychosocial model help us understand and address COVID pandemic-related disparities? And how can health disparities be incorporated into biopsychosocial research?
So I'll jump in, acknowledging that I certainly don't have a complete answer to that. But kind of my thought is that the social part of the biopsychosocial model doesn't get as much attention, and that addressing the social part, obviously disparities is a key part of that. And I think even here tonight, we haven't talked as much about the social piece and the absolute power that addressing social connection can have on our health and our well-being. And if we think about the fact, I think one of the most striking things I found in my research is how much my research participants haven't thought about their social relationships as a determinant of health. It's not something that gets talked about as much until the pandemic, when we were all having to physical distance, and suddenly the conversation was, wow, my relationships are so important, not just because they're fun, but because they're essential to my health and well-being. And so if we really want to embrace the biopsychosocial model at our visits with our primary care doctors, we should be talking about social relationships. And we're not doing as much of that, including here. So I think, yes, addressing social determinants of health, including social connection, social isolation, all of those types of things, if we really integrate that more into our clinical care, we could have a big impact on many disparities.
Well, don't
I
you
think, if I could just add, I think Kim makes some good points, and maybe this also circles us back to the issue of more sensitive periods. I think it's no accident that by many indicators, adolescents and young adults are struggling the most in terms of mental health during the pandemic. The phase in life when developing social connections, it's kind of what the brain is and what the body is designed to be doing, and expanding your social world. And so although I think we're all very cautious about this, rates of suicide among adults seem to have gone down modestly, perhaps in 2020, 21, but not the case with adolescents and young adults, and signs of depression and anxiety disorder seem to really be going up. So just another issue there.
Don't you think, though, Kim, that we haven't even seen the tip of the iceberg yet in terms of what the pandemic has done to us all? I mean, I had a friend who said to me a couple of weeks ago, I forget how to talk to people. I mean, very sad.
It is. I think also, though, if it can start us thinking about it and talking about it, that's huge. I think the fact that we've all had this intervention that taught us that we can be intentional about our relationships is massive. I think in a lot of the interventions that help people reduce loneliness or get more connected, that's a key part. Just this information that, yes, my relationships aren't just this passive thing where people call me or my spouse makes an arrangement. Like, no, you can actually plan, make goals around relationships. And so, yeah, I think it's definitely hurt a lot of us. And I think some communities have definitely been hurt more. But I do look forward to us hopefully continuing the discussion around actively addressing social connection.
So the biopsychosocial approach would be to say, even in a pandemic, there is differential exposure according to psychological factors. And just this month or recently, there was a big analysis of data from the UK Biobank showing depression predicting prospectively hospitalization for infection. So these are all sort of pointing to this important social connection as a important mollifier of illness.
I can speak a little bit to Alzheimer's disease and cognitive disorders, and we're anticipating that we're going to see more of kind of cognitive disorders that have been harder to classify, but we also expect in the long run that we may see more Alzheimer's disease. So people that have had symptomatic COVID-19, and especially those that got more sick, they actually develop at least temporary dysregulation in beta amyloid and tau. And there's worry that these things can actually seed themselves, so they can, initiate the process that might happen later, but now is triggered to start earlier. This is what has often been talked about, for example, in post-concussion, et cetera, and that we're seeing possibly something similar to that with those that received, that got COVID and survived. So I do think that then if you look at basically the change in kind of social interaction, and the fact that for many people, I think that the habits of social isolation that they kind of picked up during the last 2.5 years, they may not, revert easily. I think that there will be a lot of people that will lead a more socially isolated life, and that those things will have an impact on your development and some of the things that we worry about in late life, be that depression, cognitive disorders, overall, acceleration of aging.
One important point to keep in mind off of what Anton was saying is that it doesn't seem like loneliness increased for most older people. So what Peter was saying, it's actually the young people who struggled mostly with loneliness, but then coming back to the full idea of biopsychosocial, some older people did struggle, and it was the ones in poverty, and it was the ones who didn't have the ability to use technology. And so that's where the loneliness increased in older people. And so folks who already have some of those risk factors for dementia, we have to be most concerned about. But on the whole, older people tended to do a lot better during the pandemic and already had aging related strengths in socio-emotional development and relationship satisfaction. So want to keep in mind the strengths that come with aging as well.
That's good news.
We move in and out at these different levels. Maybe that's a chance to come back to you, Peter, and to say that it sounds like what you're saying is that social networks actually kind of operate as almost an independent entity, as an organism, as it were, influencing health, independent from their impact on each individual within it. Is that accurate?
Yeah, I think that's a good metaphor in a way. I think one of the fascinating parts of social network science in the last couple of decades has been recognition that many of the influential factors about networks really aren't evident until you're able to really see the network as a whole, that you can't detect these things through individuals' own perceptions of the network that they're a part of. One example of that, I had mentioned in the brief remarks I gave that we studied the structure of networks, including peer friendships and students' connections with adults in a number, about 40 high schools, and we found that having connections with adults was protective. It was associated with lower likelihood of having suicidal thoughts and behaviors. But being a part of a peer group where others were connected to adults added protection. And having that kind of intergenerational, if you actually shared a strong adult connection with someone you were close to, that added additional protection. So yeah, the principle really that many of these network factors are so-called emergent, that they're really evident when they come together, when they can be really observed through measurement or through assessment like that. And and so it's exciting. I think there's a lot more for us to learn, particularly about how are networks malleable. When are they malleable, and how can we leverage those effects for health and equity and other things?
Ooh, fascinating. Kim, you work in health systems as well. What opportunities are there in a health system, do you think, to better understand and incorporate, in particular, psychological and social dimensions of health, illness, and the care we need to provide at URMC and URMedicine?
Sure, such an important thing to think about, and it makes me think about our institution's strengths in patient-reported outcomes and how we are such a leader in the country around collecting those outcomes, that we're collecting depression screening across all of our clinical services. And some of our clinical services are already collecting data on social function. So what an opportunity we have to do very brief biopsychosocial assessments. These measures that are developed using item response theory take less than a minute. They usually take about 30 seconds to complete. So you can get kind of a full biopsychosocial assessment in a minute and link it to EPIC. So what an opportunity we have to really study this at scale in our health system.
Which is a network of its own, no doubt. Okay. One other here for you, Steve. You're about to open a clinic for young people at ultra high risk for the development of a psychotic disorder here. Is it really possible to prevent schizophrenia?
Yes, well, that's the goal. That's the goal of many of these clinics all over the world that use a similar model. The best data we have as of last year, a big meta-analysis, indicated that at least over a 1 to 2 year period, you can cut the rate of new cases of a psychotic disorder down by about 40. The problem is we don't know, let's say 3, 4, 5 years later, if the people still are staying healthy or how many are developing a psychotic disorder at that time. So we know we can delay it, we can prevent it in the short term. What people seem to think is that even if you can't fully prevent it, but if you can delay it by, let's say, from when someone's 17 to, let's say, when they're 20 or 21, that person just has so much more in the way of life skills and achievement and et cetera to have a better outcome over the long term. So I guess the shorter answer would be the preliminary evidence seems to be that we can delay and maybe prevent new cases of schizophrenia. Also, having people involved in treatment early is a big help in case they do develop it. And the outcomes tend to be good, relatively good there. So it's an ambitious project for sure, but we need to do more than we used to do, which is just, wait. Like you don't wait till someone has a heart attack to think they have heart disease. You try to intervene early. That's what this is, but in a psychiatric population.
Very good. We're really coming to the end of our time. There are other questions here that I'm sorry we're not going to be able to get to, and they're very challenging ones. And a question about where's that one? It's terrific. That many of you knew, several of you knew Drs. Engel and Romano. Given that you're researchers, what do you think they would say needs to be some of the primary clinical applications of the biopsychosocial model, and how does research get us from the bench to the bedside as it relates to actually making people well? That's a very broad, important conversation. I think that we need to take up in all sorts of clinical service settings here, and continue to do what we do as a major research university, with a major, very, very large clinical operation, that is to continue to draw the links between all the wonderful stuff that you are doing and the biopsychosocial answers to the illnesses that people enter our doors with every day. So I want to thank you all for being here. I want to thank the panelists for your insights and for sharing your work. I want to thank you, the audience, for enriching the discussion with your questions. Also to thank for all of us, Zena Schuber and Steve Fasone and their team for organizational and technical support extraordinaire. And ask everyone to keep in mind that there are more semi-sesquicentennial events coming along. I think Zena put one of those, the next one of these, into the chat. Take note of that, and also please visit the department website for a set of resources from the recordings that have already been made and the future events. So with that, let me say again, thank you, and good night all.
Thank you, Yeates.
Thanks, Yeates.
Bye, everyone.